and also Bachelor in Arts in Women's Studies from UC Riverside. She then joined Olive Finn's lab for her PhD. She graduated in 2017 with a PhD in chemistry. She is currently the academic coordinator in the UC Davis Metabolomics Center. Her research interests include compound identification and dereplication in metabolomics, mass spectral library development, developing method for polyphenol analysis and natural products. Dr. Vaniya has also taught international metabolomics courses since 2015. Today, she will talk about metabolomics databases and libraries and some data analysis. Let's welcome Dr. Vaniya.
Thank you, Dr. Chen, for that wonderful introduction. I'm very happy to be here. Again, thank you for inviting me to speak. I'm very excited about the topic, so let's get started. I'm going to share my screen here. Okay, and then.
Yep. Everything looks good.
Okay, perfect. Wonderful. I'm just going to put this over here. So again, welcome everybody. I'm very happy to be here. I'm from the UC Davis West Coast Metabolomics Center. My name's Arpana Vaniya, and I'm the academic coordinator. As Dr. Chen very well, described about myself, I got my PhD in chemistry from UC Davis, and my research focus and interests have been widely on creating mass spectral reference libraries, particularly for plant compounds, natural products, but also teaching in a professional education setting. So I really do enjoy doing something like this. To get started, I wanted to kind of cover our objectives that we will cover, or that we'll talk about today. So the first objective today is really explaining the challenges of identification of unknown metabolites. When we're talking about databases and libraries, this will kind of make sense as to why I wanted to talk about this first. And then also describe the different approaches to compound identification. Then I'll go into describing different types of mass spectral libraries, as there are quite a number of types. And then we'll learn about different types of metabolomic databases. So moving on to compound identification. Why are we so fascinated with the unknown? So this is kind of a central question as to what drives our research. It could be exploring space, the deep, vast unknowns of space. It could be understanding if we're really alone out there in the world, or it could be simply wanting to know what's on the other side of the moon. So there's this quote that I really think encapsulates this idea of why we're fascinated with the unknown. And it really goes about, expanding what we already know. So with this, thinking about the unknowns of our, of our lives, or, the earth around us, science in general, when we think about metabolomics, the unknowns are actually the central dilemma in metabolomics and a huge bottleneck. So when we break down the different omicses, genomics contains 4 bases, proteomics has 20 amino acids as a foundation, and metabolomics has, more than millions of chemicals. So when we think about genomics, proteomics, and metabolomics, the chemical complexity increases as we, enter the realm of metabolomics. So this is what causes the problems of, having so many unknowns to be our central dilemma. And normally in many data sets that we analyze here at the Metabolomics Center, 80% of compounds are unknown. So it's a huge bottleneck that we deal with every day. When you think about why there are so many peaks, or why there's so many unknown compounds, you might picture a, very simplistic pathway map like so, but there's actually many compounds that are coming from damage repair metabolites, for example. There are epimetabolomes to be considered. There's also lots of food metabolites that we are consuming or exposed to on a daily basis. There are trillions of microbes that, many researchers also investigate. And then there's also xenobiotics in terms of exposome. So even though we're thinking about small molecules, these small molecules have a really big impact. So when thinking about, again, these types of metabolites, the breakdown of these metabolites is categorized into known and unknown metabolites. And it's not as clear cut as just, what's known and what's unknown. There's actually known metabolites, and these are metabolites that are known and routinely identified. A researcher or a scientist can look at data and very quickly assess that, hey, this is something that I've normally looked at. It's glucose because it has these identifiers for me to know that, because it's something that we routinely identify. There is something called unknown knowns, and these are unknown at the beginning of the analysis, and then they're revealed to be a known metabolite post-investigation. This is a term called structural dereplication. So, it might appear as it's unknown, but once you do some data analysis, with the libraries that we'll talk about or the databases that we'll talk about, they're actually a known metabolite. And then you have unknown unknowns. So these are truly unknown metabolites that we're all, trying to identify in some way or another, and really try to find them in our biological studies and research questions, because this is where, the true interesting things lie in terms of some of the research questions that a lot of people are asking. So this is just kind of to keep people engaged. I want to know, how do you know if it's an unknown unknown? Just if you have time, just put it in the chat. Do you search it for a SciFinder? Do you search it for mass spectral libraries, or search it for in a database? Or if there are other methods that how you determine if it's a truly unknown unknown, go ahead and put that in the chat, and we'll continue on with the lecture here, and we'll get to those answers in a bit. Okay. So the real question for compound identification is then, how do you get from an unknown spectra to a structure? So there's this middle section here where you have your LCMS or GCMS data, but it's the steps in the middle, in the center, of getting to an actual structure. So some of that approaches I'll cover in the next two slides, but the main idea is using structural dereplication, and that's the process to determine unknown knowns, like I discussed a slide ago. So really, you want to do a very exhaustive search in libraries and databases. So normally what you would have is MS1 data, and you're collecting MSMS data, usually by collision-induced dissociation. And then you would take that MSMS data and do an exhaustive search in many libraries and databases. And this image here doesn't accurately capture the number of libraries and databases that are available to any metabolomics researcher currently. So this is kind of the normal first step, as you would say, but there are different approaches to compound identification, and I want to talk about those right now. So the first one is mass spectral library search. So this is spectral interpretation using reference mass spectral libraries such as NIST and MONA, and I'll talk about several other types of libraries as well in a second. And then we also have database searching. So this is spectral interpretation using compound databases or other databases. You might have heard of HMDB, which is a very popular database, or MINE as well. And some other techniques for compound identification is using in silico software for compound ID. CFM ID is one of them, is a very popular one that I teach quite routinely. This is spectral interpretation using hybrid rules. So this is a combination of machine learning plus bond strengths. There's also another tool called MS Finder, developed by Dr. Hiroshi Chugawa. It's a spectral interpretation with chemical reaction-based rules, specifically hydrogen rearrangement rules. Then you have Mass Frontier, which is a commercial available tool by Thermo. It's a wonderful tool that I've used in the past. And it does spectral interpretation also by chemical reaction-based rules, but it's determined all of the fragmentation rules and approaches and mechanisms based on literature. So, the developers had looked over, thousands of papers and really collected anything in literature that would point to a mechanism of some sort of fragmentation and have put it into a software, which I think is really, really helpful when you're trying to learn about how the fragmentation is occurring in your spectra. Then there's also another tool, CSI Finger ID. It's also in combination with Sirius, but spectral interpretation is done using machine learning methods plus comparison of fragmentation trees. And then there's also the use of ab initio first principle methods, or really quantum chemistry, to provide spectral interpretation. So these are some approaches and some tools, but really it's taking an algorithm, whether it's a quantum chemistry or heuristic approaches, machine learning. We learned about some reaction chemistry methods, such as hydrogen rearrangements, or those from literature, to really produce some of these smart algorithms to interpret your MSMS data. But what we're here to talk about and learn about is the mass spectral library search and database searching that takes place in metabolomics research. So first, I want to cover mass spectral libraries. This is something that's close and near and dear to my heart, as this is, has been a focus of mine for a very long time. So there are some common types of spectral libraries. The first one is commercially available libraries. These are ones that you have to pay for. Normally, common ones are NIST or Metlin. There are also freely available libraries such as MONA. I'll go over much greater detail about MONA in a second. And then there's in-house only. So these are libraries that, haven't been shared with the public, and you're using it very specifically in your research question or in your lab or your, workflow as it goes. And for those of you who do have in-house libraries, I, strongly encourage you to share them with the community at large, because that's how we're going to grow and keep moving forward. So the type of spectra in these libraries are most commonly MSMS spectra, but it's not limited to MS1 or even MS to the N. That is something that I've, worked on in the past of, creating an MS to the N library. So it's not limited at all in those regards. And then there are other types of libraries that I kind of wanted to highlight here, and those are MZRT libraries. So this is retention time of a reference standard, as well as the accurate mass precursor MZ value that's recorded from a chromatographic method. So it's very specific to the type of method that you're using. And really, an MZRT library that I create can't routinely be shared as easily as any MSMS libraries, unless you adopt the same chromatography method. Mass spectra can be accompanied with these libraries, but it's not mandatory. So in the chat, feel free to let me know what kind of mass spectral libraries you've used in your research, and I'll be reading those questions and answers at the end of the lecture. So again, the most common approach to compound ID is MSMS spectral search. This is a gold standard for metabolomics, and it's really spectral interpretation using reference mass spectral libraries and databases. And I've listed a few there. So the workflow here is ideally you're acquiring MSMS data from any one of these instruments. It could be an ion trap or a triple quad, Orbitrap. But nowadays, a lot of the instruments are more high res, accurate mass. So you do have better and better data as we progress forward in, the developmental stages of mass spec. So that MSMS spectrum or spectra are taken, and both the precursor is matched in a database, as well as the product ions or the fragment ions are searched against a database, resulting with some annotations and a report that normally has some scores to kind of help a researcher justify what is a good candidate versus what is to be ignored. So here again, this is a very routine software. It's a NIST MS search. The software is actually free. It's the library that costs money, and I'll talk about that in a second. But it's one of the oldest, softwares that we've used in the community or in metabolomics to do the MSMS searching. So what you can see here, if you can see my mouse, you can see an experimental MSMS spectrum at the top in the red that is matched against in silico MSMS reference spectra, or it could be also an experimental MSMS spectrum there at the bottom. This type of search is called a head to tail match. So you can really see, where ions are overlapping and where there are ions that, are miss and hit, hit and miss ideally, and this impacts the score. So with that, there's some pros and cons to everything that we, that we work on, or, that we are dealing with. So in terms of mass spectral library searching, there's kind of two categories and types of libraries that we are dealing with. Those libraries that are for LC-MS-MS and those libraries that are for GC-MS. So in terms of LC-MS-MS libraries, there's limited coverage in the libraries compared to the number of compounds. And I'll kind of talk about that in the next slide as to why that is. There are far fewer LC-MS libraries compared to GC-MS libraries, and just an added bonus there, GC-MS libraries are much more robust because of the technology that's used, ionization technology that's used to create those spectra. So they're more robust, and one can acquire the same compound at a different lab and produce very similar MSMS spectra, where that's kind of very difficult for MSMS libraries. For LC-MS libraries for plant metabolites, those still remain very limited compared to primary metabolites or lipidomics, for example. And then scoring spectral matches is still, in the works. We've done a bunch of research here at the Phen Lab that point to the direction that those scores that we see to kind of justify if a compound is correctly identified or not can be misleading at some points. And then creating reference libraries such as NIST or anything that you'll see in MONA requires a lots of resources. So it's time, money, a lot of patience. So that just comes from personal experience, but it does require a lot of resources and time. Okay, so I, mentioned that, these libraries have limited coverage. Even though there are many libraries out there now, we're still limited because the number of known compounds is so much larger. So in Pubchem alone, there's 110 million compounds, and ChemSpider actually just recently caught up to Pubchem, and they both have 110 million compounds. So, libraries have limited coverage of the chemical space compared to the number of known compounds. And it's really amazing because libraries like NIST, they have 1.3 million spectra, and that's only in relationship to 31,000 compounds. So it's not even like scratching the surface of what we see in Pubchem or ChemSpider alone. So just kind of jumping here, NIST is the National Institute of Standards and Technology. This is one of the types of libraries that I was talking about. This is a commercial library. And there are different types of compounds and spectra within the library. The NIST 2020 library was released last June, but it's, has been the most impressive version just due to the sheer number of spectra and compounds. Like I said, this is a commercial library. So once you have a license, you can always upgrade at a relatively lower price. And I definitely do recommend, if you're starting out or haven't considered buying this library, I really do recommend it, just because of the quality and where it's coming from, as well as, its coverage in terms of metabolites and other types of compounds. So when you're doing mass spectral library searching, these libraries will have data from different types of instruments. So it's really important to think about the instrument that you're using when you're comparing your data. So for example, if your data is acquired on a Q Exactive, you want to try to also make sure that your reference library is on a Q Exactive. But that's not always the case because of the limitation in terms of spectra that we have available. So I wanted to kind of show some cases here. So in case one with quinidine, we have a pretty good match, Q Exactive matched to a QTOF spectrum. And you can see here with the dot product, let me move over my mouse, with the dot product and reverse dot product. So these are two very common ways of scoring MSMS matching. They're both high. So this kind of indicates, that the spectral match is really good. And you can even see with your own eyes here, like how the ions are overlapping and that they're matching quite nicely. So even with different instruments, it's possible that you can get a really high score and a very good match. But in case two, this is for the same compound again on a Q Exactive at the top and a QTOF on the bottom. You can see that the energies are quite different. So one thing to note is that the way that the fragmentation occurs does differ slightly, even though everyone is using CID or HCD, there's a little bit of differences to account for. So this compound Floritin has a very low dot product score and a very high reverse dot product score. So this is the misleading portion of what I was talking about earlier, how these scores can be a little bit misleading if, a novice researcher is looking at these scores and they see, oh, well, my reverse dot product score, or they filtered, their sheet by reverse dot product score and everything is very high. But when you actually look at this, the match itself, it's very poor because there's really only one dominating peak that matches and everything else does not really match that well. Okay. And then the last case is a case of isomers, and really a huge problem when you're dealing with mass spectral library searching. So this is a flavonoid glycoside isomer. You can see that they're not the same compound, they're isomers, but what happens with isomers is that they produce very high scores again, because like compounds or like similar compounds behave similarly, and that means like as well as in fragmentation. So the MSMS that's produced for these isomers are going to be very similar to each other. Therefore, you're going to have a false positive in the sense that your match is not good, even though the scores indicate that they're good. So what do we know about dot product and reverse dot product scores? So this is a receiver operating characteristic curve at a ROC curve between iTree, which is an older library of mine, and MONA plus NIS 17. So it's just comparing the libraries against each other and to show the false and true positive rates here. So a ROC curve, just as a reminder, is a trade-off between sensitivity and selectivity. And the closer the curve follows the left-hand border, so up here, kind of where this green arrow is, the more accurate the test is. So what we can see here is that when you only use reverse dot product score, that tends to lead to excessive false positive rates. And when you kind of rely on your dot product score, or as it's shown in the graph, a cosine score, you're going to have a lot more true positives. So, when you're analyzing your data, just, be mindful of the scores that you're using, or cutoffs that you're using for these scores to kind of justify those candidates lists, and always visually check your MSMS matching. Okay, so moving on. And now I want to talk about MONA. So MONA is a mass bank of North America. It's a freely available metadata-centric auto-curating repository designed for efficient storage of mass spectral records. So this is a library or really a database that's developed here at the Fiend Lab. And what we're trying to do is bring all mass spectral libraries together in a way that's well thought out. It's supported by a lot of metadata. There's a lot of auto curation that's happening before it's even made public. So there's a lot of steps that go into making this publicly available. And just taking a look at our progress over the years, we've really increased in the number of total spectra, MSMS spectra. We're almost at 700,000 spectra here in 2021. It's not over yet, so we still have time to bring more libraries into MONA. And really the cool thing about it is you can download and select spectra as you like, right? So that's something that's really amazing. We have databases such as HMDB in MONA, GNPS, Mass Bank. So we are really trying to bring everything that's available, publicly available, into MONA. So this is something that I was talking about in terms of the metadata that's available. So any one of us can really upload spectra. It's quite easy. It takes a couple steps. You just have to, register, make an account, and upload spectra. I do this quite frequently, but this is what it looks like on the end for the user. So you'll see information that they've provided in terms of method information, the retention time, the columns that they use. So really you could essentially create such spectra in your own lab if you gather the same materials that's needed. But you could also just download this exact MSMS spectra if you also need it as well. And it's really easy to download. That's one of the things that is really important that we achieved in MONA, is how easy it is to download, because that's not the case in a lot of some other libraries and databases that I've come across. And one thing I want to point out is that all spectra that are uploaded by others are, given authorship and all of that stuff. So everything is always listed by the name of the author who submitted it and all of that. So yeah, it's been a really good growing project and something that I'm quite passionate about. So just kind of wanted to show you how easy it is to download. So you would just go to, you would simply visit www.massbank.us and click on downloads to access the mass spectral libraries and any or all spectra in MONA. And that's, like I said, close to 700,000. So here on the downloads page, you'll kind of see some categories. In the upper portion of the page, you can download all spectra. You can download in silico spectra only, experimental spectra. There are further breakdowns of positive versus negative. And then there's some additional downloads if you are a programmer that need, the spectral or chemical identifiers for all MONA records. So not only is these libraries good for data processing and doing mass spectral library searching against your experimental data, but it's also really beneficial for the cheminformatics side of metabolomics, or researchers who are, coding and creating software, and they need benchmark data sets. So it's really nice to have access to such vast amount of data from different instruments, different compounds, and different libraries. And then lower, when you kind of scroll down on the page, there's a section where all the libraries are available. You can see that there are quite a number of libraries there. We've added a couple new ones. EMBL MCF was an endogenous metabolites and lipids library that was recently added. And all the ones that have a leaf next to it are plant metabolites that are specifically covered in those libraries. Nowadays, you'll see the crossover, like there are plant metabolites in HMDB, for example. So you'll see kind of that crossover happening, but these libraries like Respect, GNPS, Metabobase, the Vaniya natural product library, they originated as purely secondary metabolites or natural product library. So again, you can see even in this list, those are limited compared to the greater scheme of things in terms of just primary metabolites and endogenous metabolites or lipids, for example. So GNPS is also a place where someone can access mass spectral libraries. GNPS stands for Global Natural Product Social Molecular Networking. And though it does exist in MONA, you can also use GNPS on its own. So it's an open access knowledge base data repository for community-wide organization, which enables sharing of process data, identified data, library data, reference data, in the community. So it's really community driven, which I really, really appreciate. So why use GNPS? You might ask yourself. You have open access to data sets. So if you are trying to learn about metabolomics or practice on some data sets, you do have access to those data sets and can download them. You have open access to spectral libraries, like I mentioned. There's high throughput of structural J replication. So going back to a couple slides from the beginning, I mentioned that structural J replication or an exhaustive search against libraries or databases is really the first step to compound identification. So this is something that can be taken care of in GNPS. And then it also has an added benefit of doing molecular networking for visualization. And I'll have a quick slide on that. And there's something called living data by continuous analysis. So this is something unique to GNPS. You can submit your data, overnight. Let's say you're a grad student, and in the morning, you'll get a text message or an email notification saying that, hey, we've identified 10 compounds, and you can go take a look at your identifications. So it's really nice. And as long as the data is in your account or on the database, it's continuously analyzed. So maybe a year later, you've published the paper and have forgotten about that data set, but you might get an email notification again, hey, we've identified a true unknown or something like that. That's kind of like the big picture idea of this living data and continuous analysis. Okay, so the construction of molecular networks is one approach of using the MSMS libraries in GNPS. So what one would do is they would input several MSMS spectra. Those MSMS spectra are simultaneously searched against the libraries in GNPS, and then similar MSMS spectra that are your experimental data are also aligned so that there's no duplicate spectra. Then those matches from the library matches, as well as the overlap of, or I guess removal of duplicate spectra, are created and merged into a network. So what you see here in this gray node, this is an unknown from your data. This other gray node is another unknown, but it's aligned and it's in a network with this compound as a surfactant, C14. So because these nodes, that's what the terminology is in a network, the dots are called nodes, which are representative of your metabolites as well as your MSMS data. They're connected to this known compound. You can, assume that there's some similarities between this unknown and this known compound there. Okay, so moving on. When you think about all of these MSMS libraries, and I haven't, covered all of them just due to the time and all of that, you kind of have to come to this question of quality versus quantity. Do I use all of the libraries available, or do I only use like what's really good quality? And this is not to preface that any of these categories are poor quality, but when you're talking about having like the highest match in terms of MSMS data as well as MZRT, you really want to get to that top tier level. So quality kind of decreases as you're going down this pyramid, but it doesn't mean that any in silico library that you use is lower quality, and I'll talk about why I say that. So the highest quality in my opinion is when you have reference standards that are run in your lab, and you've acquired not only MSMS data, but you've also acquired accurate MZ and retention time information. So you can see here that with those types of metadata, you're going to also increase your confidence. So the higher the quality of data, the higher the confidence that you have when reporting these identifications. Then there's also a category where you have the same gradient and you have the same instrument, but the data or the reference data is acquired in a different lab. So it's not exactly the same, which might propose some differences, or maybe some retention time are shifting, but that can be taken care of with corrections if you have internal standards in your methods. So again, pretty high quality, but not as best when you run it in your lab. Then there's data that's coming from the same instrument, but you have no retention time. So again, earlier we talked about, matching MSMS data from different instruments, how sometimes it can work and sometimes it can lead to false positives or lower scores, depending on the compound and the energy that's used to acquire that MSMS data. And then you have completely different instruments, which just kind of falls into the same category that we were talking about, but you have no retention time information. So you can see as we move down this pyramid, we're losing more metadata and more lines of confidence in terms of that annotation or that identification that we're trying to make at the end of the day. And then you have in silico generated spectra. This means that there's no instrument or no complete set of compounds. There are in silico libraries that are made off of a few reference standard experimental data, and then the program or the algorithm that's used is learning from the patterns that they're observed in that small set of compounds, but it's not a complete set of reference standards. So it's not that it's poor quality. It could be very high quality library. It's just the lack of trust, I feel like, might not be there for everyone. Recently in 2019, for example, Metlin had removed all of their in silico data from their library, and it caused quite a stir in the Twitterverse. Some people agreed with that decision and some people didn't agree with that decision. So, even with in silico MSMS data, there's a little bit of conversation to be had in terms of whether we want to trust it or not. And then there's no match at all with any library. So things to remember in this case, any of these libraries or types of libraries can have different coverage of compounds, and they can be at a different level. Even though you have library data from the same instrument, maybe it's only for plant metabolites. These library types are parallel to each other. They're not sequential, so you don't go like one after the other. They're just, you know, the types of libraries that might be present or that you might use. And then at the bottom of the tier with the no match, when there's no matches, you could use in silico tools for confirmation, which is highly recommended. A lot of these in silico tools that I briefly mentioned are freely available, and they have tutorials on their websites, very easy to use as well. So some quick shout outs, other mass spectral libraries for metabolomics. There are several from Bruker, for example, Metabobase libraries. There's a personal library that you see next to Metlin, which is actually a collection of Metlin for Bruker instrument users. And then there's Metabobase plant library, which is available in Mona. There's also the HMDB version of Bruker metabolite library. There's WiseMass MS library, there's MZ Cloud. I also found this SW drug mass spectral library, which is EI-MS Spectra, but still freely available. Wiley Registry is known to have many mass spectral libraries. This is one of the newer ones, the 12th edition, but they have different categories for compound specific libraries. But those are commercial libraries. There's one from India called Tandem Mass Spectrum Database. You can view the data, but you can't download the data. So that was something that I mentioned as one of our goals in Mona was, you know, having that accessibility and downloading it quite relatively easy. I also found one, Tomkits. This is a freely available library of 426 MSMS spectra covering 213 opioids. That's freely available. And Cayman Chemicals also makes EI-MS Spectra library, which was something new to me that I learned. But some of these libraries, or most of these, are all downloadable and free, with respect to a few that are not. Okay, so moving forward to databases. Just want to make sure how I'm doing on time. Okay, so a few more slides and we'll be wrapping up this lecture here. So we've talked a lot about libraries, and now I'm moving on to databases, but I really wanted to answer the question, what's the difference between them, right? So a database is an organized collection of data that's stored and accessed electronically from a computer system. Databases can be easily accessible, manageable, and updated on a end use, you know, like not a user's side, but the programmer's side. Whereas a library is also a collection of reference mass spectra used to identify molecules, but libraries are not limited to experimental data only. They can have in silico mass spectra. In my opinion, databases are more fluid, whereas libraries are static. They don't change as much. Again, databases are easy to update. Mass spectral libraries are not so easy to update. And in most cases, databases are freely available, where this is not always the case for libraries. However, in metabolomics, the term database and libraries have been used interchangeably in the community. So, you know, when I started out as a novice, you know, grad student, I was always confused about this, but you learn quickly that, oh, people are actually referring to the same thing. HMDB, the Human Metabolome Database, is something that is very popular. It comes from the Wishart Research Lab, and it contains detailed information about metabolites found in the human body. The database was first created to link 3 types of data, which is chemical data, clinical data, and molecular and biological biochemistry data. So it does this very well. For an example, fumaric acid here, you can have so much information at the tip of your fingers, whether it's the description of the metabolites, the synonym, the ontology, the physical property. There's even spectra that's been embedded, because a lot of these databases do collaborate with each other. So you'll find, you know, Mona spectra in HMDB, or you'll find Spectra that's in Mona in Pubchem. So there's a lot of crosstalk between a lot of these databases and libraries. And then there's links to other databases. So something that I really appreciate about HMDB, or really the Wishart Research Lab, is they're expanding metabolomics databases vastly. So there's just like a database for almost everything. Just alone on their website, they have over, I think over 20 databases, anything from drugs to foods to, phenol, explorer, bovine rumen metabolome, so yeast metabolome. They have a lot of databases that you can use and kind of explore. And then in the HMDB resources page, if you have time to look over that, there are over 30 databases that are listed across 6 categories. So some of the categories are drug, comprehensive metabolome, disease and pathology, compound. So this is kind of really impressive in terms of the types of resources you have accessible to yourself. Dictionary of Natural Products, something that's not on that list that I wanted to mention, is a compound database. So it includes chemical, physical, and biological properties of compounds. It's been updated recently and it looks great. It's very easy to use. You just search by name or by structure, and you can get all the identifiers or information about the compound. There's no spectra in the database. It's just purely information about a specific compound. The blood exposome database is a compound specific database. So it covers chemicals in the blood. This was developed back in 2019 here in the Fiend Lab by Dr. Dinesh Barupal. And again, the idea is similar to the fact that you can find information about blood or chemicals in the blood, the blood exposome. So there's over 65,000 unique compounds, but the database was created so that you could rank chemicals to build your own libraries, associate blood chemicals or compounds with specific phenotypes, get detailed descriptions about chemicals, prepare a list of blood chemicals by classes, interpret your data set, so on and so forth. So it's very easy to use. Again, highly advise that you check it out and play around with it. Then there's another database called Kegido Encyclopedia of Genes and Genomes. This is a metabolic pathways database. So you can really start to look into different pathways. For example, the citrate cycle, TCA cycle, something that we're maybe all familiar with. So you can search different pathways, and also in different tools, you can then take those pathways and link them with your data and show things that are upregulated or downregulated. There are tools to do that in your data analysis portion. I don't have time to talk about that, but if you're interested, please email me and we could definitely chat about that. Binbase is a GCMS metabolome database developed here in the Fiend Lab. It's a database that covers 8,000 metabolites from over 1,600 studies, which is close to 110,000 samples. But this isn't really accessible to users until the creation of Binvestigate. So this is a web application that helps you explore the Binbase database. It's a really cool tool that allows you to kind of look for GCMS spectra based on specific species or organs. For example, here I looked for cancer cells in cancer cells and found that there's more than 1,000 bins or 1,000 spectra that might be found in cancer cells. So it's really cool. If you have an unknown spectra, you can search this in Binvestigate. And the last thing I kind of want to hit on is how can you communicate across different databases? Okay, so I've talked about a bunch of different databases, and all of these databases have different IDs. So if, as a researcher, if you wanted to crosstalk between these different databases, there are options to do that. One is called CTS, Chemical Translation Service. And this allows you to convert in a simple conversion, that's a one-to-one conversion, or even do batch conversion relatively quickly. So it converts between common identifiers. Maybe you wanted to convert from a chemical name to an INCHI key because that's what the journal that you're publishing in has a requirement, but your list is like 1,000 and you don't want to do that manually. So you can just submit this to CTS and it'll do that quite quickly. And then there are different websites and database IDs that you can translate between. There's also Pubchem Identifier Exchange service. This also does something very similar, but it's focused around Pubchem CIDs. Something that's unique to this is it can convert between INCHI keys to SMILES or vice versa in the other way around. These tools or services is something that I routinely use almost all the time. And so with that, I'd like to go over some summaries and key takeaways. With the summary being that MS library matching is the most common approach to compound identification. It's routinely used in metabolomics, but the coverage of chemical space is still limited. So we saw how Pubchem and ChemSpider, they have 110,000 or million compounds, and we only, in some of the databases have so much spectra. So it's not even reaching the point of, covering the known compounds that we know about. Scoring for spectral matches is not perfect. So if you routinely use reverse dot product only, this could lead to more false positives in your results or your end product. There are 3 types of libraries: commercially available library, freely available library, and in-house libraries. Again, if you have any in-house libraries and would like to share them with the community or upload them to Mona, please reach out to me and I can help you with that. Definitely think it's good for the community and good to share those reference spectra that might be unique to your lab. Databases versus libraries. Databases are much more fluid. They contain vast amount of data, and they're just easy to search against and update and all of those things. And then translation. So CTS and Pubchem ID Exchange can be used to kind of translate identifiers and communicate really between those databases. So with that, thank you for your attention. I'll take any questions. If you have questions later down the road, feel free to email me. We do offer courses as well. So yeah, with that, I'll take questions and that there.
Thank you so much. There are already several comments about how excellent this talk is. A lot of great information in the chat. But there are also some really good questions in the chat. During your talk, you did a couple of inquiries, but I only see a couple of people replied. One is Rose Stephen, I think, uses MS Finder.
Mm-hmm.
Shweta replied she used MZ Cloud.
Oh, that's awesome. Yeah, I've used MZ Cloud too, but it's not something I routinely use, but I definitely agree. Like MZ Cloud is has a vast number of spectra and lots of compounds, and they're constantly developing it. So it's really excellent. There was a question here. In terms of MS Finder, do you mean hydrogen rearrangement or hydrogen gas rearrangement? Mm-hmm. It is hydrogen rearrangement, Rose. So just to clarify that. And then
Great.
There was a question from Lloyd: during mass spectral MSMS matching, can you compensate for difference in instrumental platforms by varying collision energies, i.e. collect tandem spectra of unknowns at various energies and search all in the attempt to find the best score? Yeah, you can do that. Routinely what we do is try to collect data at 10, 20, and 40 to kind of have a coverage of medium, high, and low. And then that's also really nice way to tackle some of these in silico software, like CFMID predicts high, medium, low spectra developed from QTOF. So we do that. A lot of the libraries that I develop, I try to hit at least 35, 45, 65, depending. So I get a variation of fragmentation. And then depending on how you create your method, that spectra, MSMS spectra at the end could be an average of those collision energies that you've used, or you could separate them out. Just depends on how you make your method. And then there's another question: what proportion of MONA has accurate mass tandem spectral data? Let's see, go back, it might be easier. I think a majority of the spectra are accurate mass. There are a specific one here. Okay, there's just the button. Like the Vaniya phi natural product LTQ, that would be, low resolution spectra, but anything from Q exactive, QTOF, the instrument tag or metadata is available. So that's something that would indicate if it's accurate mass or not. But some of the libraries do indicate that. Bilal had asked if this recording will be available later, and I believe that's yes.
Yes.
And then another question from Lloyd: at what point do you recommend the validation of identifications obtained via external databases matching using orthogonal technologies such as co-analysis with authentic compounds or NMR? I think it really depends on how your statistics play out. So a lot of the time when I'm teaching, compound ID strategies or using these software, people ask me like, well, if I have 80% of metabolites that are unknown, do I submit everything? And there are techniques and there are ways that you can survey your data and submit everything to some kind of analysis like that. But I think the one idea that I've always, had in my head is that you need to kind of have a statistical approach first to kind of narrow down that list of what's important, to really then go into NMR and go down that rabbit hole and try to figure and identify at a high MSI level, to say the least. And then any advice on CEU mass mediator for searching metabolites in different databases? I'm not familiar with CEU mass mediator, so if you could explain what that is, maybe I might have an answer for that, but I'll keep continuing with some questions. Where do you think CCS data would fit in the quality versus quantity slide? That's a great question. So I think that's something that I don't talk about at all with iMobility and CCS data, but I think it would definitely, let me go to that slide. 28. I think it would be kind of towards the top, because again, what I was trying to hit with this slide was that the more metadata that you have that's closer to your experimental data in terms of how you acquire that reference data is going to be better in terms of providing higher confidence and providing, a correct identification or annotation, to say the least. And I think, okay, so how do you further validate putative identification of metabolites with a cosine spectral score of less than 0.7 in GNPS? Honestly, I think I would, my approach is to try other tools as well, other than GNPS. Now that, MONA is easily downloadable, searching against MONA to see how your experimental spectra would turn out is a good idea, or searching in different tools is also a good strategy. You can take that individual spectrum and search it against MONA. You don't necessarily have to download the entire library as well. So I think when you have something that's not as strong of a score, taking it outside of what you're working with and seeing how it fares in those softwares or libraries. Could you suggest more databases as accessing mass bank seems not possible over here? Oh, okay. So Musa, we could connect later and I could help you on how to access Mona and kind of help you with that.
Yeah, so
And
Oliver just commented, is www, just massbank.us.
Oh yes, that is correct. That's what I had on my slide. Thanks,
Okay.
Oliver. And then I performed lipidomics on my samples, but many were annotated lipid maps to metabolites which were not present in plants. Should I report it as unknown? Have you used other types of libraries, I guess, is what I would kind of ask in response to that. Again, we can connect privately. I'll stay on the chat. And we can kind of address those questions. And that's the end of the questions on the chat. Again, thank you for your time. Thank you, Dr. Chen, for inviting me to speak. And Musa, I can help you on the chat here, and we can have a private chat.
It's still not showing up on that website. Well, thank you so much. Really appreciate and great talk and really nice discussions. I think the time is perfect. So this is the end of this session, but we do have another session in 15 minutes. So please stay on or come back. And we have great talks in the afternoon. We have a great discussion sections to address any remaining questions. And we're gonna hear from leaders, and I will invite all the speakers to join us in the discussion session as well. So please stay on and have a nice 15 minutes break. And Dr. Sally Osman will chair the afternoon session. Thank you. Thank you to all the speakers, and thanks everybody for your attention. See you in about